As we have discussed about the complement system, now we are going to discuss about the immune responses. Although this topic is not very much important from exam point of view, but it is important to understand this topic to understand the mechanism of immunity in our body. So let's start with the classification of the immune response. The immune response is of two types. One is the cell mediated immune response and the other one is the antibody mediated immune response. Now it's quite clear from their name itself that cell mediated immune response is mediated by different types of cells in our body. Like examples, for example, we have cytotoxic T cells, NK cells, or the antibody dependent cellular cytotoxicity, while the antibody mediated immune response is mediated by the antibodies which are secreted by the plasma cells. We will be discussing about these two types of immune responses in quite detail in our upcoming videos, but here one should be very much clear that these two immune responses are not separate entity. They are interdependent to each other. That means the cell mediated immune response requires the help of antibody, while the antibody mediated immune response requires different types of cells for it to act. Okay, so these two are interdependent. This feature of interdependency you will understand better when we will be discussing about these two types of immune responses in detail in upcoming videos. Now, irrespective of whether it is a cell mediated immune response or antibody mediated immune response, two events are always occurring there. Okay, this is irrespective of whether it is CMI or AMI. CMI for cell mediated immune response and AMI for antibody mediated immune response. What are those two events which occur is irrespective of whether it is going to be a CMI or AMI? Those two events are: number one is the antigen presentation to the helper T cell, while the second event is the differentiation of that helper T cell into type 1 or type 2 helper T cell. Okay, now first of all we will be discussing about the antigen presentation, how the antigen is presented to the helper T cell. Now, for the presentation of the antigen, there must have been some dedicated cells which can engulf the antigen and then present this to the helper T cells. What are those cells? Those cells are the antigen presenting cells. Those are examples are the macrophages, the dendritic cells, and the B lymphocytes itself. So these three cells are the dedicated antigen presenting cells which present the antigens to the helper T cells. Now there are some other antigen presenting cells also, but we will be focusing over these three mainly. Okay, the dendritic cells are present in skin, lung, and GI mucosa. You can remember this also. Now, how are they going to present? So they engulf the antigens into themselves, and then what do? They process the antigens. Okay, and what happens after processing? Due to processing, the antigen is broken down into multiple smaller antigenic fragments, and those antigenic fragments are loaded over different types of MHC molecules over the surface of those or the over the cell membrane of those antigen presenting cells, and then uh, that antigen fragment is presented to the helper T cell. Now there are two pathways of presentation of the antigens or the antigenic fragments. Those two pathways are the cytosolic pathway and the endocytic pathway. When how does the antigen presenting cell decide whether it is going to take the cytosolic pathway or the endocytic pathway? That is dependent upon the type of antigen. If the antigen is a intracellular antigen, if it is a intracellular antigen like viral antigen or tumor antigen, then it will be presented over the MHC1 molecule of that antigen presenting cell to CD8 T cell, which is also called as the cytotoxic T cell. Okay, this is the cytosolic pathway. It is used for intracellular antigens. It is used for the intracellular antigens. It is used for intracellular antigens which are either viral antigens or the tumor antigens. While if the antigen is a exogenous antigen, then the endocytic pathway is taken. If it is a exogenous antigen, then the endocytic pathway is taken. How does this get presented over the over the antigen presenting cells? So the endocytic, if the antigen is processed through the endocytic pathway, then it will be presented over the MHC2 molecule of that antigen presenting cell, and it will be presenting to CD4 T cell, okay, not the CD8. So remember, when it is taking cytosolic pathway, then the presentation is done over the MHC1 molecule, okay. When it is taking cytosolic pathway, then presentation is being done on the MHC1 molecule to the CD8 T cell, okay. But when it is taking endocytic pathway for the exogenous antigens, then it is presented over the MHC2 molecules for CD4 T cells, okay, CD4 T cells, that is the helper T cells, okay. So this can be remembered by simple fact that CD, if you multiply 8 with MHC1, then it comes out to be 8, and if you multiply CD4, the 4 of CD4 to MHC2 molecule, then also it comes out to be 8. That means you have to keep the multiplication of these two things to be 8, okay. And then by that you can remember that the cytosolic pathway uses the MHC1 molecules for presentation of the antigenic fragments, while the endocytic pathway uses the MHC2 molecule for the antigenic fragment presentation to the helper T cell, that is CD4 T cells, okay. Now what are those two, you must be wondering, what are those two MHC molecules? So MHC molecules are normal, molecules present over the normal cells, out of which the MHC1 molecule is present on all the nucleated cells plus all the antigen presenting cells also, because those antigen presenting cells are also nucleated now, so they are present in all nucleated cells. But the MHC2 molecules are present only on the antigen presenting cells, only on the antigen presenting cells. So please remember that MHC2 molecule is present only on the antigen presenting cells, while MHC1 molecule is present on all nucleated cells. What inference can we derive from this information? So we can derive from this information that the any normal cells even which is tumor infected, they can also present their antigenic fragments over MHC molecules to CD8 T cell, okay. But this helper T cell activation can only be done by the APCs, not the normal cells, okay. This helper T cell activation can only be done by the antigen presenting cell, while the cytotoxic T cells can be activated by the normal cells which are viral infected or which are which have tumor in them. So those nucleated cells can uh, activate cytotoxic T cells by MHC1 molecule, but the MHC2 molecules are present on the APCs, and the APCs can activate the T cells only, helper T cells only, okay. Now we will be discussing how the T cells or the helper T cell gets differentiated after the presentation of the antigenic fragment to it. So see here, what is happening, that the processed antigen loaded over the MHC2 molecule of the APCs binds to the T cell receptor of the CD4 T cell. Now this single signal is not enough for the activation of the activation of the helper T cell. It requires also the CD28 on the TH helper T cell to bind with the B7 of the antigen presenting cells. So CD28 molecule of helper T cell also binds to the B7 molecule of the antigen presenting cells. Along with that, there is also cytokine signal. Along with that, there is also cytokine signal, which is interleukin 1 acts on the helper T cell. That interleukin 1 is secreted by the antigen presenting cell, so these three signals are required for the differentiation of the helper T cell. What happens after that? After these three signals are received by the helper T cell, then this helper T cell gets activated and starts secreting the interleukin 2. That interleukin 2 acts on same helper T cell, plus it also acts on other helper T cells as well. As a result, there is formation of the memory cells plus the effector helper cells. Both will be produced, the memory T cells and the effector T cells. Now that effector T cells can be either the type 1 helper T cell or the type 2 helper T cells. If it had been the type 1 helper T cell, that means it is going to activate the cell mediated immunity, while if it is a type 2 helper T cell being produced after the activation of the helper T cell, then it is going to induce the antibody mediated immune response by different types of cytokines. Now how is it decided whether it is going to be a type 1 helper T cell getting produced or it is going to be a type 2 helper T cell getting produced after the T helper cell activation? So this is completely dependent upon the cytokine or the interleukins which are present in that media. Okay, so the in the environment, sorry. So depending upon that cytokine or the interleukin which is present in that environment during the activation of the immune response, depending on that, the either type 1 helper T cell or type 2 helper T cell is produced. Now if it is a type 1 helper T cell, then it will be producing the interferon gamma, the TNF beta and the interleukin 2, which will cause the induction of the cell mediated immune response, while if it is a type 2 helper T cell getting produced after activation of the helper T cell, then it will be producing interleukin 4, 5, 6 and interleukin 13, and this will cause the activation of antibody mediated immune response. So these are the two things that we see irrespective of whether it is going to be a cell mediated immune response or it is going to be a antibody mediated immune response. These two events always occur in case of immune response development. So this is all about the first part of the immune response. In the next part we will be discussing the cell mediated immune response and the antibody mediated immune response.